Good afternoon, everyone. Today is the fifth day of our training program, and our second eminent speaker for this afternoon is Dr. Sanjeev Kumar Tripathi, sir. Dr. Sanjeev Kumar Tripathi, sir, is working as an assistant professor in.
So, please, there is a correction. My name is Sanjeev Kumar Prajapati, not
Sorry.
Tripathi.
Dr. Sanjeev Kumar Prajapati, sir. Dr. Sanjeev Kumar sir is working as an assistant professor in Department of Hydro and Renewable Energy at IIT Roorkee. Before joining as a IIT Roorkee, he served as assistant professor in chemical and biochemical engineering at IIT Patna during May 2017 to June 2019, and as assistant professor in biotechnology at NSIT Delhi during August 2014 to May 2017. Dr. Sanjeev Kumar did his BE in biochemical engineering from Kumaon Engineering College, Uttarakhand in 2008. After obtaining M.Tech in biotechnology from Anna University, Chennai in 2010, he pursued a PhD in environmental biotechnology and bioenergy from IIT Delhi in 2015. During PhD, he worked towards exploring and integrating algal biomethane production with wastewater treatment. His research was published in several international journals of high repute, including for his outstanding research contribution, he was awarded with the prestigious distinction in Doctoral Research Award from IIT Delhi. Currently, he is the principal investigator of the Environmental and Biofuel Research Lab at IIT Roorkee. His research group is working on various aspects of bioprocess technology in the areas of energy and environment. So today we are very fortunate that Dr. Prajapati sir accepted our invitation and joined us for this training program. And today he will discuss on the topic challenges and opportunities of biogas from algal biomass. So on behalf of the National Agriculture Project, Dr. Panjabarao Deshmukh Krishi Vidyapit Akola, I welcome you sir for this training program. And now I request you to start your presentation, sir.
Thank you, Dr. Swati, for the nice introduction. So, should I switch off my video? I think it will be good.
Yes, sir, no problem.
Yeah.
Yes.
Yeah, am I audible?
Yes, sir, you are audible.
Okay, great. So, let me share the PPT first. Yeah, is this slide visible?
Yes, sir, it is visible.
Okay. So thank you, participant, for attending this talk, which will be interesting, I believe, because it's related to the energy recovery from the algal biomass. And I am thankful for the organizing committee for this wonderful opportunity to share my work with the different organization and the colleagues working in the different sectors. So, the topic of talk is challenges and opportunity of biogas production from algal biomass. So the lecture will start with the brief introduction about the the algal strains. So what the algae actually are, because this is not the common term which is used for the algae. So some people might not be having some idea that what is algae. So I will briefly start about the algae. Then I'll talk about the some of the prerequisite requirements related to the algal biogas production, which should be carried out. And then I'll discuss some of the key finding of my work, which has been very productive in terms of new development in the area of algal biogas production. So I'm starting the lecture with the description of, before starting, please note down my email ID if you need some further query, which we may not discuss or we could not discuss in this meeting. So you can send me a mail on this email ID, and I will be happy to provide a detailed description of your query. Okay. Yeah. So first thing is that before talking to the biogas, we should know what is the algae. So these are very simple organism, or we can simply call them as a miniature plants, because of the compound which is known as chlorophyll, which is found in the algae. So like plant do have chlorophyll and they can do photosynthesis, the algae can do similar thing. But the difference is that most of the algae, like microalgae, are of miniature size. They cannot be seen through the naked eye. Okay. So otherwise, fundamentally, these are related to the plant, but as these are very microscopic in nature, these also belongs to microorganism. So some of the features include like a simple miniature plant, and they usually require water to grow. And sometimes if there is a moisture, sufficient moisture for their growth, they can easily grow. And like plant, they do photosynthesis, means they can convert CO2 into organic matter. The chemical reaction which is derived with the help of solar light. And most of the algae can be found in terms of single cell, means the individual cell can grow independently, and most of the algae can grow as a cluster, and some of the algae which are known as macroalgae are multicellular, means a single algae will be having number of cells to make a single organism. Like macroalgae, one example of macroalgae is kelp. And I'll talk more about the growth pattern of the algae, but timing we can consider that one of the common mode of algal growth is photosynthetic. Otherwise, the algae can also grow mixotropically or heterotropically. So I'll briefly discuss about the mixotropically, heterotropically in future slides. And so far, as per my understanding or the data which I have with me, around 36,000 species of algae have been identified. And these all algae usually have chlorophyll or other pigment which are used in the photosynthesis. And in broader term, the algae can be divided into different classes. Some of these are including the red algae, which is a macroalgae, green algae, some of the green algae are macroalgae and some could be the microalgae. And the term macro and micro, if you get confused, one is the macroalgae and second one is the microalgae. So micro means their size is around in micrometers, because means you cannot see them through the naked eye, particularly when you're talking about the single cell, but you can see a growth or a colony of algae through the naked eye. But macroalgae are visible and these are larger in size. So they are having multicellular organization, whereas microalgae are single cells. So usually they are microalgae are singular cells. Okay. So the another class is the brown algae, which are the seaweeds. So most of the macroalgae are also called as seaweeds. And some other algae are diatoms, blue-green algae, and the dinoflagellist. So this blue-green algae, it's a very interesting microorganism because it's not a true algae. So because these are similar to bacteria, but most of the algae are not similar to bacteria. The blue-green algae are similar to bacteria. But the difference is that these are a special kind of bacteria which are having the chlorophyll. So other algae will be having the chloroplast. Like plant have chloroplast for a photosynthesis, but these blue-green algae do not have chloroplast. Instead, they have the chlorophyll in their cell wall, which is used for photosynthesis. Otherwise, structurally, all the blue-green algae are bacteria. So some authors report the blue-green algae or the cyanobacteria along with the algae itself, but fundamentally, these are a different class of bacteria which contains chlorophyll. So this is a basic idea about the algae. So different classes of algae like macroalgae and microalgae, you can differentiate them into different classes based on your their structure, their distribution, and their growth pattern. These can be classified into different groups. So the fundamental mechanism behind the growth of the algae is the photosynthesis. So that's why these are considered as more superior than the other microorganism when we talk about the treatment of wastewater or their growth, because if we talk about the wastewater treatment, sometimes we have to supplement the carbon sources. But in case of algae, as they can do the photosynthesis, and usually they are phototrophic in nature, so they can take up CO2 from the atmosphere. So there is no need to supply carbon material for their growth. But some algae, if the carbon is available, they can utilize the organic carbon or they can directly take up the CO2. Okay. So the mechanism is same, which is similar to the photosynthesis which is happening in the plants. Okay, so this is the simplest representation of photosynthesis. So this could be of two kind, like oxygenic photosynthesis and anaxogenic photosynthesis. So I'll briefly explain the difference. Like usually when we write the photosynthesis, we consider the CO2 as the main feedstock, and with the help of water and the solar energy, it is converted into glucose primarily. Then glucose is metabolized in the cell structure within the cells, and then it will convert it into different biomolecules which make different component of algal biomass. But as you know, the biomass also contain nitrogen and phosphorus and different other elements. So this is not the accurate stoichiometry of the photosynthesis. Rather, there should be some terms which should include nitrogen and phosphate also. But in simple way, we can simply say that CO2 is being fixed with the algae and it is being converted into carbohydrate. This is the simplest way. So this happens when the water is acting as a electron donor. Otherwise, if the water is not available to act as an electron donor, so this kind of photosynthesis, because there will be no oxygen generation as a result of photosynthesis, is known as anoxygenic photosynthesis. Okay, so what actually happening here? The algae is taking water as a medium and the carbon dioxide from the atmosphere and the sunlight, and now it is converting into carbohydrate, which are then metabolized within the cell itself and then stored into different components like starch, oil, and cellulose, and proteins also, which is missing here, proteins. So mainly the biomass of algae will be having the lipid or oil, protein, and carbohydrates. So these two are starch and cellulose, both are carbohydrate. I hope you all know what are the carbohydrates. So basically, any kind of biomass, including the algal biomass, will be having the three major kind of macromolecules or the biomolecules, which are known as lipid or oil, proteins, and carbohydrate. So this is something which makes them a very attractive feedstock for biogas production and different other biofuel production. So this is brief about the photosynthesis, though there are very in-depth mechanism available to understand the photosynthesis, but whenever you go into depth, you will get more confusion if you are not having some background. So I limited this to a very simple way so that everyone in the lecture and those attending this lecture through the other platform can also understand. But still, if you have some queries, you can ask me. You can drop me a mail. I will be happy to provide the answer to your queries. Yeah. So when we talk about the biogas production, first thing we should know that for producing the biogas, we should have sufficient feedstock. And in this particular case, the feedstock is the algal biomass. So first thing should be how we can produce the algal biomass, which will serve as a feedstock for biogas production. So there has been significant work in the direction of different configuration of photobioreactors. The reactors which are used to cultivate the algal biomass at larger scale, so that we can get sufficient biomass for production of biofuel, including biogas. So some of the pictures are shown here, like tubular reactors are there, some thin film reactors are there, polywave reactors are there. So there are different configurations which are available and can be used depending upon the requirement, depending upon the end use of the algal biomass which is being produced. So these are few common type of reactor which are used in the lab scale as well as industrial scales. And these two systems which are shown here, I'm just clicking them. So this particular system is the attached growth system which was developed at IIT Delhi during my PhD. So this is another kind of reactor. All these systems which you can see in the first part of the picture are suspended type. But these are the attached growth system, and there are different kind of attached growth system available, as you can see in the literature reviews. Okay, so these systems are very productive in terms of biomass production, and because I tested them, I know that we can harvest the algal biomass just after 48 hours to get sufficient biomass. So these systems were developed and then scale up to a 200-liter capacity. And these another kind of system which are known as raised bed ponds, which were also developed at IIT Delhi during my PhD itself. So these are portable kind of raised bed ponds, and you can take them as per your requirement. You can relocate them, you can install them, and once the work is done, you can remove them from that place. So the use of these system is not only for the production of biomass, but also in treatment of wastewater. Okay, and the simple mechanism of the raised pond is like that. There is an agitator which will agitate the fluid, and the fluid keep mixing throughout the or circulating throughout the system, which makes the algal cells into suspension, and they are exposed to light, and they are interacting with the nutrients present in the media, and then they can grow at higher rate. So these are some of the common type of photobioreactor which are used for algal biomass production as well as wastewater treatment. So reactor, bioreactor, and photobioreactor, these three terms are used when we talk about the algal cultivation. But the main difference is that all the photobioreactors are bioreactor because they include some biological components. And because we are using the light, either solar light or the artificial light, that's why these are known as photobioreactor. Yeah. So coming to the cultivation type, because algae can grow in the closed system like tubular reactor, or algae can be cultivated in open system like raceway pond. The example is HRAP, which is high rate algal pond. Okay, so the selection of different kind of cultivation system will depend on the final and ultimate use. For example, the algae, apart from the biofuel potential, they also have several metabolites. Okay, so metabolites are having the application in food sector, pharma sector, cosmetic sectors. So there are several applications of algae apart from the biofuel production. Okay, so if our aim is to produce the metabolite, we usually will go with the closed system because we can cultivate the pure culture very easily in the closed system. We have the control, we are having the control flow of liquid, we are providing proper sterile condition, means we are avoiding the contamination by the different microorganisms which are present in the air. So usually when our interest is to produce high value metabolites, or if we are not worried about the cost of algae cultivation, in that case we can go with the closed system. But particularly when we are talking about the algal production for the biogas or some other biofuel, in that case open raceway ponds are good choice, because we are not worried about the contamination, and mostly the algae will be growing, and it will be easy to control their growth. Sorry, it will be difficult to control their growth, and sometimes the contamination, if it is very dangerous, may create some problem. But still, when we compare the economy, we are not interested to waste much money into the cultivation system itself. That's why usually when we are focusing on the this biogas production of the biofuel production, we usually go with the open system type. And these two systems are having different advantages and different disadvantages. Like few are listed here. In case of closed reactor, the operational cost is very high. And yeah, so these are having some advantages like high rate of CO2 sequestration, which are not possible in the open recipe pond. But the added advantage of open recipe pond is that low operational cost, economical for biofuel production, mass scale cultivation is possible. Otherwise, if you are using a tubular reactor, you have to install multiple tubular reactor. In case of if you are producing large scale biomass for biofuel production. But in case of raceway pond, you can easily construct a raceway pond and you can use the open cultivation system. Okay, so these are two main kind of system. One is the closed one, in which mainly the pure culture will grow, and second one is the open type, in which usually the algae which is growing will be the mixed type. Means there are chances that there are different kind of algae growing at a time, and there may be some bacteria which are growing in this energy with the algal strains. And the choice of cultivation system is always decided based on the final end use of the grown biomass. Yeah. So some of the advantages which have been listed earlier, and I'm discussing here again. So like high photosynthetic efficiency and fast growth rate when we compare the algal growth with the terrestrial biomass. That's why they are having the improved land use efficiency compared to the terrestrial biomass. And you can cultivate the algae around the year, provided in winters you have to maintain some temperature conditions. Otherwise, you can grow the algae throughout the year. Okay. And these can be grown in the wastewater and non-agricultural land. Means there is no competition with the crop cultivation. If you have some waste land available and you have the wastewater resources, you can use them for algae cultivation. And these algae are having very high rate of CO2 fixation, the another added advantage of the algal system. Like the algae is the CO2 is the only carbon which usually use the algae cultivation. So CO2 from the atmosphere can be recycled and fixed into biomass. And one study says that the rate of CO2 fixation is around 1.83 kg CO2 per kg of dry algal biomass. So what we are doing, we are minimizing the greenhouse gases in terms of CO2, and in turn we are treating the wastewater and we are getting the biomass produced for the biofuel production. And also the recovery of nutrients from the wastewater is possible, because the algae, when it will grow in the wastewater, they will take up nitrogen, phosphorus, and some other micronutrient. They will accumulate in the cell itself. And when you separate the cells, you can use the biomass as a slow release fertilizer. Either you can go for the biofuel production, or you can use the grown biomass as a slow release fertilizer. But interestingly, the algae is having very high content of biodegradable compounds. That's why one of the most nowadays, I can say, the popular substrate for biogas production. Yeah. So one thing I mentioned, that algae can be grown in the wastewater, provided the wastewater is free from some toxic compounds. Sometime if the metal is present, or some toxic pesticides or some other chemicals are present, in that case, the growth may be suppressed, or the algae may die in the wastewater. Otherwise, like sewage wastewater, which we have having, or the municipal wastewater, or the industrial wastewater, which are not having the toxic compound, can be used for algae cultivation. So the advantage is that the wastewater can be transferred to the reservoir ponds. So what we are doing, we are fixing the CO2 from the atmosphere, and we are treating the wastewater, and in turn we are getting the algal biomass produced, which are having different biomolecules for conversion into biofuel. In turn, we will get the treated water, which usually, as per my understanding and my experience, is at par with the discharge standard suggested by CPCB. So after cultivating the algae in the wastewater, we can directly discharge to the nearby water body. The treated water can be discharged, or it can be used in different other applications like agriculture practices, or the horticulture applications. And I hope you will be having a question in your mind, that how will algae work in wastewater treatment system so efficiently? If you have heard about the bioremediation, in those systems, the microorganism, usually bacteria or fungi, are used to treat the wastewater. And sometimes we have to supplement the carbon source. For example, if we are treating the sewage, which are not having the high carbon content, in that case, to support the microbial growth, we need to supply some carbon and some nutrients to support the growth of microorganism. So that these are some limitation with the bioremediation. But this particular sector, which is known as the phycoremediation, it's a branch of phytoremediation. Phycoremediation, in which we are using the algae, and we need to add the organic carbon, because algae can take CO2 from the atmosphere. The CO2 will be dissolved in the water, which will be taken up by the algae. And the fundamental mechanism behind the algal role in the wastewater treatment is that the synergy between the algae and bacteria. If you can see in the picture, if we are growing the algae, bacteria, which are aerobic in nature, so bacteria will need O2. Means if you are treating the wastewater in a reactor using bacteria, in that case, in the wastewater, you have to supply the air. But in this case, when we are using algae, bacteria will also grow. Those are the native bacteria in the wastewater. So what will happen? They will make exchange of CO2 and O2, because bacteria need O2, and these will degrade the organic matter present in the wastewater and will release CO2. So CO2 released by the bacteria will be available to the algae, whereas the algae during the photosynthesis will release oxygen, which will support the growth of the bacteria. So it's my personal observation, when I cultivated the algae in the sterile wastewater, the growth was some value. But when I cultivated the algae in a unsterile wastewater, the biomass production was very much higher, and the treatment efficiency of the unsterile wastewater was quite high. This is just because these algae and bacteria are growing together in the wastewater system, and they are making the synergy through the exchange of different nutrients and the CO2 and oxygen. Apart from this, the algae will release some O2 to the environment, and CO2 from the atmosphere can also be taken by the algae. And the biomass produced can be taken into the biomethanation plant, or the anaerobic digestion plant, known as AD, which will result in production of biogas. Okay, and there are several develop technology by which you can remove the CO2, because the biogas will be having around 60% of methane, which is having the fuel value, and around 30% of CO2. These value may change depending upon the feedstock type and the conditions, operational parameter of the biogas plants. But usually, roughly, we can assume 60% of methane and 30% of CO2. And rest will be the H2S and some other gases in trace amount. Okay, so CO2 is not having any fuel value. Methane is having the fuel value. That's why we have to enrich the biogas, particularly when we have to use the biogas into transport sector, for example. In that case, we have to remove the CO2. So there are several technology by which we can do the enrichment, and we can remove the CO2 from the biogas, and we can get the methane-rich biogas, which is having around more than 90% of methane. And the CO2 from this process can be recycled to the algal cultivation system. So if you are doing this, means you are recycling the CO2 to the algae cultivation system. Obviously, more CO2 is available, the algal growth will be more better. Okay, so this will improve the chances of very high rate of treatment and very high rate of biomass production if we are integrating the system. And as a part of the process, you will get the solid digested part, which can be used as a fertilizer, and the liquid part from the biogas plant can be recycled to the algal cultivation system to support more biomass production, because the algae need nitrogen and phosphorus, which will be released during the anaerobic digestion. It can be recycled to the algal cultivation system. So this results in a complete closed system, which is actually releasing the energy only, and everything rest is recyclable. Means part of these things we are using the system itself, and part of the product like solid of the anaerobic digestion process can be used as a biofertilizer. So that's why I consider the algal-based biogas production is a very attractive, and I believe that it will be one of most popular kind of biofuel in the future, because now the government is more focusing on the CBG, the compressed biogas. And there are several list of feedstock available for the CBG production, and very soon the algae will be there in the list, because algae has huge potential for the wastewater treatment along with generation of biomass, which is a very perfect substrate for biogas production. Yeah, so apart from removing the nutrient, there are few reports, not based on my personal experience, but there are few reports which says that pathogens from the wastewater can also be removed from the wastewater. That's why one of my PhD scholar is looking if we can remove the pathogens from the wastewater using the algal cultivation, and the high total nitrogen and phosphorus removal rate can be obtained, and these system can be operated at decentralized manner. Means there is no need to make a centralized unit. We can have a small unit at different location, and we can do. And obviously GSG reduction is a added advantage of this, and we are getting the biomass as a biofuel precursor. Yeah. So when we talk about the integration of the algae with the wastewater treatment plant, which are existing in India itself, so in that case, there will be two possibilities. First thing is that we are having the effluent, the wastewater, it should be screened, and after primary settling, it can go to the algal reservoir pond, provided the COD or BOD levels are low. Means the organic content or the pollutant level is very low. Mainly nitrogen and phosphorus there. For example, in case of sewage, in that case, we can directly take that particular water into algal cultivation system for treatment and biomass production. But if the COD or BOD is high, in that case, we can include the algae as a polishing stage in the existing sewage treatment plant. Means the system will be based on the treatment of wastewater based on the activated sludge process, for example, or the existing technology which is there in the sewage treatment plant. Then we can link the algal reservoir pond to further polish the treated water, because sometime the treated water will be having the high nutrient content. So the carbon can be removed in these two stages, like activated sludge can remove the organic carbon, but the nutrient removal potential of the activated sludge process is relatively low. In that case, we can polish the water with the algal pond, and we will get the biomass produced, which will be available for biogas production. So in gist, we can say, if we are interested in algal biogas production, we should focus on the low cost production of biomass. If there is a huge production cost, that may not be acceptable at industrial scale. That's why there is a search of alternate growth media In that case, wastewater seems a suitable growth media, and I have been working on this system. Like I have worked on different kind of wastewater to be used as a growth media for algae cultivation, and we are treating the wastewater, which is the added advantage, and we are getting the biomass, which is available for biofuel production. So this is how the algae can be integrated with the wastewater treatment plant just to improve the energy recovery. Yeah, so you can estimate the potential, like in Indian scenario, the sewage generation is around 30 mld, 29,000 mld. Okay, but the installed capacity is around 6,000 mld. So around 22,000 mld of this untreated sewage is being discharged to the environment. Okay, so which is having different pollutant like CO2 is there, nitrogen and phosphorus is there. If we are discharging these water untreated sewage into the water bodies, obviously there are several environmental concerns. Okay, so when we are trying to expand our sewage treatment facilities, so particularly if we compare this with the activity sludge process, so roughly for treating 1 kg of BOD, we require 1 kg, 1 kilowatt of electricity for aeration. And this produces around 1 kg of fossil CO2 from power generation. So you can see the disadvantage of the process. But if we are using the algae for the treatment of wastewater, so for 1 kg of BD removal by a photosynthetic oxygenation, provided we have both algae and bacteria in the system, so no energy input is required because we are not doing the aeration. Some amount of energy will be required because sometimes we have to mix the system so that there is no settling of algae. And this will produce enough microalgal biomass to generate methane, which can produce 1 kilowatt of electric power. So you can easily theoretically compare the algae mediated treatment system with the activated sludge process. Okay, so that's why I believe that very soon the algae will be there in the sewage treatment plants to replace the existing treatment setups. Okay, yeah. So some advantages includes like cost-effective, low energy requirement, short harvesting, which I have discussed earlier, and some reports says that it's 100% removal of E. coli and other bacteria at pH 9. So the algae, when grown in the wastewater or in any media, it shifts the pH towards alkaline. So this alkaline pH usually kills the pathogens. So these are some of the advantages or the possibilities which should be explored, particularly when we are talking about the algal biomass as a feedstock for biogas production. Because without having the sufficient amount of feedstock, we cannot produce biogas. So these slides, some of you will feel that these are not related to the biogas production, but these are crucial because until and unless we have the biomass produced, we cannot produce the biogas. And for production of algal biomass, we need growth media, and that can be easily replaced with the help of wastewater. So my intention to cover the wastewater related slide in this lecture was just to ensure that the cost of algal production is very low, particularly when we are using the waste stream for cultivation. Yeah. So once we have cultivated the algal biomass, there are different routes by which we can convert the algal biomass into biofuel. Like there are thermochemical conversion routes, there are biological conversion routes. So through the biological conversion route, we can produce the biodiesel, biohydrogen, bioethanol, and biogas. And today we will be talking about the biogas production. Okay, so these are several other options which can be done, but usually in case of thermochemical conversion, the biomass should be dried. Okay, it should be dewatered and dried. Then we can go for the thermochemical conversion. But in case of biological conversion, no such requirement is there. We can use the wet biomass instead of drying it. So obviously the cost of conversion will be relatively less. Yeah. Now the question is, if we are having a range of processes which we can use for converting the algal biomass into biofuel, then why only AD? So this is the question which initially was in my mind when I started working for my PhD at IIT Delhi, that I can go with the algal biomass, I can produce the biodiesel. During that time around 2009 or 10, the biodiesel was one of the most popular biofuel. So I thought that why can't I go for the biodiesel production and I'm going for the biogas production? So answer will be clear when we discuss this slide. So there are different methods like gasification and direct combustion, but these include high input cost. Usually dewatering and drying is required. Okay. Less energy recovery, and there is a possibility of release of toxic gases through the combustion. Again, if we deselect this particular option, then we have the biodiesel production. So again, in case of biodiesel production, high input cost is there just because before the diesel production, we have to extract the lipid. And lipid extraction include the drying of the algal biomass and dewatering it. Okay, then there is again input of cost and energy. So and then it has to be converted through the chemical process into biodiesel. The lipid has to be converted into biodiesel through chemical process, which will again require some catalyst and energy. So I deselected this one, and I thought of biohydrogen and biomethane production because these two requires wet algal biomass. Means no need of drying the algal biomass, no need of dewatering it, no need of pre-processing. Okay, so that's why the cost of cost input in the process was limited. And the added advantage which I discovered through the literature review were related to this. The wet biomass, one advantage. Energy recovery per unit biomass compared to biodiesel and direct combustion was relatively high theoretically based on different papers I could conclude. And there is a possibility of coupling different biofuel route. For example, you have algae. Okay, you extract the lipid, take them into the biodiesel production, then residual biomass can be taken for biogas production. Similarly, you can take the carbohydrate out of it, carbohydrates, and then take carbohydrate for ethanol production. Ethanol production, and the residue can be go for biogas production. Okay, and similarly, you can club the process with the metabolite recovery. Okay, so there was possibility to doing the coupling with the different biofuel routes and leading towards the biorefinery approach. Okay, so this is the another advantage of anaerobic digestion process, and the resulting flux of ammonia and phosphate as a part of the liquid digested, which will be obtained after the anaerobic digestion process, can be used as a nutrient organic fertilizer. And instead of this, what I try to do, I'll show you in some slide that the liquid part was the AD slurry was separated into two parts, solid and liquid. And liquid was then recycled to the algae cultivation, you can see. So the nutrient from the biogas plants were recycled to the algae cultivation for the further growth of biomass, which resulted in an integrated closed loop process. Okay, so I found that the production of biogas from the algal biomass, instead of going for other biofuel production, was quite interesting in terms of low energy inputs, low cost input, and higher recovery of energy per unit of biomass. Yeah. Again, when we talk about the biogas production, the question is that there are several feedstock which can be used for biogas production. So why algae? Why not some other feedstock which are commonly used? Like there are several plants which are working on the chicken manure and cattle manure or pig manure. Okay. So similarly, the agro residue are also used for biogas production. But when I compare the theoretical methane potential of the algal biomass with the non-algal biomass, I found that the theoretical methane potential, which is the SMP in this case, is stoichiometric methane potential. I'll tell you how you can calculate this. So I found that the methane potential of the algal biomass is significantly higher than the non-algal biomass. So then I thought this is the right choice. So I should go with the algal biomass for biogas production. Though these systems are running properly, and there are lots of plants which are coming with the which are based on the different feedstock like agro residue or the cattle manure. But still, algae has advantage that we can grow the algae in the wastewater treatment system itself, and the biogas production potential is relatively higher than the non-algal biomass. Yeah. So now I'm coming to the fundamental of anaerobic digestion process, because before going to the application part, we should know what actually happened in the reactor itself. So fundamentally, we should know, because sometime as a plant operator, we can see that biogas production has stopped. So we should be able to answer that what are the key process parameter from where the process has been stopped. So we can take up the sample from the anaerobic digester tank, we can analyze it, and based on the analysis and understanding of the mechanism of the process, we can comment that this is the faulty process. And these are the limitations which have, this is the particular point where the problem has occurred, because of this, the biogas production has stopped. So that's why we should know what are the mechanisms involved in the conversion of biomass into biogas. Okay, so in principle, there are 4 stages in the reactor itself. In a single reactor, there will be 4 stages. When we feed a substrate to a bioreactor, it will go through the 4 stages and finally convert into methane. So first stage is that hydrolysis. Because I told you that biomass are mixed up of carbohydrate, protein, and lipid. So the biomass include the municipal solid waste, okay, or municipal organic solid waste, more correctly. So these organic waste will be having the lipid, carbohydrate, and protein, and these all are polymers. Means they are having the long chains, and are not available for the methanogens to convert or consume them and convert into biogas. So these are very difficult to be taken up by the methanogens, which are the actual microorganism. Those convert the biomass, the organic carbon into methane. Okay, so that's why in this complete process, there are range of microorganisms which are involved at different stages. So it's a teamwork actually, teamwork by the microorganism. Some will act as a hydrolytic bacteria, which will be doing the hydrolysis of the macromolecule. You can consider it's a large ball, which is being broken down into small pieces. Okay, so the organic polymer in the biomass will be hydrolyzed by one class of bacteria known as hydrolytic bacteria into smaller molecule, which are known as organic monomers. Like sugar will be there from the carbohydrate, amino acids or the polypeptides or small peptides will be there from the protein, and long chain fatty acids will be there from the lipid. Okay, so this is the first stage in the anaerobic digestion process. Initially, the biomass or the feedstock will be hydrolyzed with the one specified group of the bacteria, which will convert them into smaller organic molecules. Then there is another class of bacteria which are known as acinogens. Means what they will do, they will take up these monomers, they will consume them, and then they will produce the organic acids, which are known as volatile fatty acids. So these volatile fatty acids along with ethanol, etc., will be produced as a part of acidogenesis. So initially hydrolysis will occur, then acidogenesis will occur. So in the hydrolysis, what is happening here? The macromolecule are being converted into monomers. Then monomers are being converted into small acids because methanogens cannot take the monomers as well. Okay, so they need help from some other microorganism which are known as acidogens. So they will convert the monomers into acetic acid. So there are different carbon chain, like up to C6 carbon chain are usually found in the anaerobic digestion slurry, which are having the volatile fatty acids. Okay, then in the next stage, there is a different class of organisms which are known as acidogens. So what they will do, they will convert all these acids into acetate and hydrogen and CO2. So these 3 are the main substrate for the methanogens. So what will happen? These acids will be converted into acetate, mainly acetate, hydrogen and CO2. Then methanogen will take or act on these 3 molecules, then convert into methane and CO2. So this is how a simple process, which looks a very simple process on superficial level, is a very complex process. And there will be lots of biochemical reactions which are occurring in the cells itself or in the reactor itself, because macromolecule are being converted into monomers, then acids, then finally acetate and hydrogen, and finally to the biogas. And the 4th stage is known as biomethanization. Okay, so these are 4 fundamental processes. Okay, so what can happen in this? For example, so I'll discuss it later on when I'll discuss this stoichiometry. So we can leave it. Okay, so this is the overall mechanism which are involved in case of the conversion of any kind of feedstock into the biogas. But particularly when we talk about the algal biomass, like as shown here, the microalgal biomass. So the scenario is a little bit different, because what will happen when we talk about the algae, okay, and when we talk about the AD process, so AD processed microbe require soluble organic. Okay, but in case of algae, all the organics are packed inside the cell wall. Okay, so means these are not available for the microbes which are there in the AD process to be converted into biogas. So they cannot take up. So again, the hydrolytic bacteria are having the ability to produce some enzymes known as exoenzymes, means they can produce the enzyme within the cells and they can release the enzyme into the environment. So what these enzymes will do, these enzymes will stick onto the surface of algal cell wall and they will partially or fully metabolize the algal cell wall. Okay. So initially what the hydrolytic bacteria are doing, so they are in search of the food. They have sensed that algal biomass can be act as a food, but they are not able to utilize it. So what they will do, their metabolism will force them to produce enzymes which can be taken out from the cell surface, because the algal cell cannot be taken by the bacteria itself. So they will produce enzyme, they will release it to the environment, into the liquid part. So these enzyme will act on the algal cell wall. This will solubilize the cell wall and the organic content will be released in the media. Now the macromolecules are available for the bacteria to act on. Now these hydrolytic bacteria will take up these compound and will break it down into smaller monomers. So this is the additional stage. Means when we talk about the cattle dung, already it is digested. So there is no need of hydrolytic bacteria to produce exoenzyme. But particularly when we talk about the algal biomass, the cell wall has to be ruptured first. So they will produce some exoenzymes. Then the organic matter will be released in the media. Once it is released, the process will be same. Okay. And there are few arrows which I have shown like this. So these are possible sites of inhibition. For example, sometime if there is accumulation of volatile fatty acids. So again, all the processes are being carried out with the help of enzymes. So enzyme involved in this process or the bacteria involved in this process can get inhibited through the accumulation of any of these products. Okay, so this is a different story, but these are related to the control of process, because sometime if the pH of the system is very low, obviously biogas production will stop. And in some cases, if the pH of system is very high in alkaline range, the biogas production will again stop, because the process is mainly based on the microorganism, and the microorganism are very specific to their proper growth conditions. And for AD, the pH is usually in the range of 6.5 to 7.5. If the range is in this range, the process will run proper. If the pH is below 6 or below 5, in that case, the process may get affected. Same will happen if the pH is going towards the alkaline conditions. So this is the fundamental behind the mechanism which are involved in the reactor itself. But at the superficial stage, we cannot see. We can see that we are adding the feedstock. It is being converted into biogas with the help of some bacteria. But in general, or fundamentally, there are a range of bacteria which are acting on the substrate, which has been fed into the reactor. Yeah, so these were the theoretical information or the bookish information which I was interested to share with you all. But now I'll focus on some of my experimental findings which I did during my PhD, or part of this is being carried out in the lab itself. Okay, so next few slides will be focusing on the work which I carried out for the production of biogas from the algal biomass. Yeah, so the story started with the cultivation of biomass. So initially, I procured some of the algae from the culture collection, like NCL Pune is there, and one culture I got from IERI Delhi, and few native microorganism or microalgae I isolated from the soil, and few from the wastewater itself. Okay, so there are different kind of algae which were used. Initially, the cultivation scale was 100 ml. Then it was scaled up to 4 liter with the different efforts from the different colleagues of mine during the PhD stage. So then we scale up it to 20 liter. Then finally we scale up it to 200 liter, and we were having such 8 systems. So total working volume was around 1600 liter. And all the time, most of the time, the algae was being cultivated in the wastewater, which we were collecting from the sewage line itself. Okay. Parallelly, one of my colleague, Pushp Shala, working on the CO2 sequestration. So our aim is to produce more biomass. So what he did, he collected some CO2 from the waste resources and tried to optimize the growth of the algae under the CO2 pressure. Okay. So this is how the process was scaled up from the algae which was there in the wastewater or in the soil, or was around in 5 ml in size. Then it was scale up to 1600 liter. Then after cultivating the biomass, these are two things which you should do, either you are working on the algal biomass or any kind of substrate for biogas production. So these are two crucial things: ultimate analysis, the elemental composition, and the proximate analysis. So one thing is that you should know the elemental composition, carbon, hydrogen, nitrogen content in the biomass, which can be done with the help of CHN and analyzer. Okay, and you should know the calorific value and the material balance. You should know. Okay, so these are crucial. I'll tell you how these, why these are crucial, and parallelly you should do the proximate analysis. Proximate analysis to know the volatile solids content, and the biochemical content you should know. And in case of algae, the cellulose and hemicellulose are of not much interest, but in most of the biomass substrate, you should do this. So you should do the ultimate analysis and the proximate analysis. Okay, so these two initial experimental will give you some interesting data related to the different kind of biomass. Like you will get the data on the volatile substance content. You can comment on the carbon content of the biomass. And more interestingly, you can comment on the CYN ratio. I'll tell you that CYN ratio is a crucial factor when we talk about the AD. Okay, if you know the carbon content, you know the nitrogen content, you can easily calculate the CYN ratio. Okay, so these are few fundamental things which are required for any kind of substrate if you are interested to convert that substrate into biogas. Okay, so this is the data which is based on the elemental composition. You should know that how the data will look like. Like elemental composition, I have tabulated here. This is published by one recent study. So you can see the carbon content, you can see the range, and according to this, you can develop the CYN ratio. Okay. So different algae will be having different CYN ratio. And sometime when we are changing the condition, for example, initially we cultivated the algae in a nutrient media. Now we are shifting to a different kind of wastewater. The CYN ratio may change. Okay. The elemental composition is very subjective to the environmental conditions, the particular type of the algae which is being grown. Okay. So that's why the CYN ratio should be calculated at regular interval. Similarly, the biochemical composition, you can tabulate like protein, carbohydrate, lipid, and volatile solids. Okay, so the volatile solid should be known because in most of the cases, the biogas production should be reported in terms of per gram of volatile solids, because different algae will be having different range of volatile solid content. So it should be compared at a same scale. Okay, so this can be done. Then how this data is useful? Okay, for example, you started working on some feedstock and you have more than 20 type of samples. Okay, let's say 20 algal strains are there in the domain, in your domain, and you're trying to work on these for production of biogas. Okay, so it won't be feasible for you to do the practical biogas production analysis for all the 20 sets, because in the research we do in the triplicate. Okay, so 20 into this will become 60, and sometimes you have to have the control, then number will increase. So obviously it's a very tedious task when you are focusing on 20 different kind of algae for biogas production and you are interested to do the practical analysis. So instead of this, what you can do, you can grow the algae at a smaller scale. Like you can get 5 or 10 gram of 5 to 10 gram of biomass. Okay, that will be sufficient for biochemical and elemental analysis. Okay, so this biomass you can do. Then after knowing the chemical composition, you can derive the empirical formula. If you know the empirical formula of any kind of biomass, you can apply this equation given by Simpson and Buswell in 1933 just to calculate the theoretical methane potential or the stoichiometric methane potential, which is the SMP, where this Vm is the molar volume of the methane, which is 22.4 liter at STP. Okay, so instead of going for the 30 days, for example, this will go for 30 days or 40 days or may go up to 80 days. So instead of this, you can quickly have some idea that which of the algae is having the high SMP value, and you can screen easily. Okay, so if you know this empirical formula, you can take this value A, B, C and put in this equation. You can calculate the stoichiometric methane potential of the algal biomass, which will give you some initial screening that what kind of algal biomass is suitable for biogas production. One thing. Second thing, you can also calculate the theoretical methane potential using this equation. If you know the biochemical composition, for example, you see that A into Cl. So A is the specific methane yield of lipid, which is very well reported in the literature. And Cl is the, you can see Cl is the lipid content in the dried biomass. So using this equation, If you know the lipid content, protein content, and the carbohydrate content, you can easily estimate the theoretical methane potential. So you have two tools by which you can initially screen the different algal strain based on their biomethane production potential. So instead of going for the practical one, you can screen out of these 20, you can take 5 or 4 or 6 up to, for example, and you can work on those for actual methane production. So this is how the elemental composition analysis, which will give you the BS volatile solid content as well as the elemental composition. So this will give you some idea that how you can screen the different algal biomass for biogas production. I hope the content is deliverable to everyone. We will discuss the queries. If you have any queries, we can discuss after the lecture. Okay. Yeah. So the added advantage of the empirical formula is that if you know the empirical formula of biomass, what you can do, you can estimate the final methane concentration in the final biogas. Roughly, you can estimate. Exactly. Practically, it may be different from the theoretical calculated value, but theoretically, you can calculate. For example, in a simplest case for conversion of carbohydrates such as sugar, for example, we are taking or starch. So in that case, an equal amount of methane and CO2 will be there in the produced biogas. You can see the stoichiometry. If you know the empirical formula, you can apply the stoichiometry and you can know the moles of methane produced, moles of CO2 produced, if the biomass is completely being converted into biogas. So in case of carbohydrate, it is 50-50%. In case of protein, the ratio is around 55 to 45%. You can see from the stoichiometry itself. And in case of protein, obviously ammonia will be there. Okay. So I'll come to this ammonia later on. In case of fats and vegetable oil, typically the ratio will be around 70 to 30. If you solve these two, you will get the ratio around 70 is to 30. So theoretically, you can predict the maximum biogas concentration in the produced biogas. So this is the added advantage of the elemental composition analysis and deriving the empirical formula. And just to derive the empirical formula, you can refer to the chemistry book of class 11th or 12th, where it has been very clearly explained that how you can derive the chemical formula if you know the elemental composition. Yeah, so far I have been discussing about the theoretical aspect by which you can screen. Then the experimental design which I followed during the biogas production experiments. So these are based on the BMP protocols. Okay, so there is a very interesting paper which I would like to share with the organizing committee so that it can be circulated with others. So it's a very interesting paper which explains the BMP protocol. So BMP is biochemical methane potential protocols. So I followed that paper, I followed the protocols, and I worked on the algal biomass for production of biogas in the batch mode. Initially, the work was carried out in the batch mode just to understand the process. So I used the algal biomass, different algae, more than 9 kind of algae. The batch reactor, initially 50 ml of BOD bottle, sealed with controlled gas opening was used as a reactor for biogas production, and the working volume was kept at 300 ml. And the rest of the volume was there just to store the gas. And the operating conditions, temperature was around 36. Substrate concentration I kept at 5 gram per liter based on the different literature reports. And inoculum to substrate ratio was kept at 3, which is also known as the food to microwave ratio. And the HRT I selected initially for 30 days. And the gas volume was measured after at 48 hours. So yeah, I forgot to mention that this is the paper which I was talking about. This is a paper on BMP protocol. So you all should read, if you are interested in the biogas production, you should read the protocols which are listed in this paper. So this is, I think, from DTU, Denmark Technical University. So you can refer this paper and you will be having the better understanding on the experimental design. Okay, so the acidic water displacement method can be used to estimate the biogas production. So this was done just to ensure that there is no mixing of the CO2 from the biogas into the water. Okay, and the net biogas production can be calculated because when we are planning some experiments, in that case, we should have some control. Control means a bottle which is having the inoculum only. So inoculum was added, but no algae was added. So this will act as a control and there will be some biogas produced from the control. So that should be subtracted from the experimental value just to get the actual value of the biogas which is being produced from the algal biomass. And then we can calculate the cumulative biogas production using this equation. So this is how I proceeded, and I screened around 9 different kind of algae using 2 which are listed as species 1 and species 2. These are the native strains which I isolated from the wastewater, this one, and this was isolated from the soil. So I compared the biogas production daily profile as well as cumulative profile. And now I found that this particular strain, SP1, is having the highest biogas production potential. But still the value was less than 250 ml methane per gram of volatile solid. If you can remember, the SMP value was greater than 600 ml per gram VS. So I was far behind than the actual theoretical value. So practically I could achieve around 250 ml. So which was significantly less than the theoretical estimated value. So I selected this particular strain to work on. Then I carried out detailed investigation on the process just to understand that what are the obstacle in the process. By the complete biomass is not being converted into biogas. If the theoretical it is showing 600 ml, I should at least get around 400 ml, but the value was around 250 ml. So I extended the period from 30 days then up to 45 days. And I regularly monitor different processes like TN, total volatile fatty acid, sugar, and pH during the process. And I found that there is no inhibition of any kind of toxic material, like ammonia is not there, total volatile fatty acid is not there. So these two you should know. If there is an accumulation of TAN, ammonia, in the reactor, so obviously pH will increase. Okay. If there is an accumulation of acids, volatile acids, the pH will drop. So I did not found such fluctuation in the pH, and there was no accumulation of ammonia, neither the total volatile fatty acids. So this was not the limitation in the process. And of course, when I extended the period up to 45 days, there was some increase in the biogas production. So I noticed that the digestion of algal biomass relatively slower. The algae is having the organic content packed in the cell wall. So this could be the major hurdles. Further, I analyzed the samples under the microscope of the digested slurry. So at 0 day, you can see there are different algae. Then at 9 day, I noticed there are intact algal cells in the digested. Digested means the content which I withdrawn from the experimental bottles. So I could notice that after even 45 days, there are intact cells of the algae which are present in the reactor. So I found that the digestibility of algal biomass is the major limitation in the process. And based on the literature, I found that the cell wall resistance to the anaerobic microbes is the one limitation. Second could be the low activity of anaerobic microbes due to the improper C/N ratio. Because in my case, the C/N ratio was less than 9. And C/N ratio for AD should be optimally in the range of 20 to 30. But if it is greater than 10, the process works perfectly. But if it is in the range of 20 to 30, the process will work efficiently. Okay, so I found that these two could be the possible reason, and I started working in this direction. So what I did, so these are related to the pre-treatment requirement, means there is a need to disrupt the cell wall because everything is packed in the cell wall itself. So I need to break this down so that the organic content can be released to the liquid part and can be utilized for biogas production. Okay, so for the treatment, what I did, I used a fungi. So I hope you all will be knowing that fungi is a good source of enzymes. Most of the commercial enzymes are being produced from the fungi. Okay, so I use a fungi which are having the cellulosic activity in their enzymes. So I use that enzyme as a crude enzyme, and I tested that enzyme for the treatment of algal biomass. So I found that the Aspergillus lentulus, one of the fungi which I tested, and the crude enzyme at 20% load, volume by volume, was having the significant pre-treatment in the algal biomass, which you can see. If the cells are intact, they will remain suspended. If the cells are broken down, due to the sticky nature of the cellular content, they will stuck and they will settle down at the bottom as a cluster. And same thing I verified through the microscope analysis, and I quantify the cell number in terms of live cells and dead cells. And I found that the fungal enzyme is efficient for the disruption of algal cell wall. Okay, then, but the problem was that the process took around 48 hours. The enzymatic pretreatment took 48 hours because I used unoptimized condition. Then I optimized the condition for the enzymatic pretreatment, and the process of pretreatment was time reduced to 6 hours. So within the 6 hours, I recovered that, I noticed that around 50% of biomass was solubilized. Means I recorded the biomass solubilization in terms of soluble COD. So this much degree of COD recovery I noticed within the 6 hours. So I selected that process. Then I took the pre-treated biomass and carried out the biogas production experiment, and I found that there is a significant improvement in biogas production. You can see here the untreated biomass having the around 255 ml per gram of volatile solid, the methane production yield, but it was improved to 324 with the pre-treatment. So this was very much confirmed that pre-treatment is required, but still the value of biogas production was low, because it should be more than 400 at least. If not 600, it should be more than 400. Okay, I consider that the AD process is not 100% efficient. Obviously, it cannot convert all the biomass into biogas, but at least this much value I should have got. But the value was around 324. Okay, some other parameter were also improved. So the another key term was that CYN ratio. So because I treated the biomass, but I did not work on the CYN ratio. So in the next run, what I did, I used cattle dung, because the cattle dung was having around 28 to 32. The CYN ratio of the cattle dung was around 28 to 32. So I used that cattle dung with the algal biomass, and I did the co-digestion with the pre-treatment, and I found that the pre-treated algae with the cattle dung, when digested under the anaerobic digestion process, the biogas yield was around 413 ml per gram of volatile solid added. So this was significantly higher than the other sets. Like fresh algae I used the fresh algae with the cattle dung. I used fresh algae with the sugarcane bagasse just to improve the CYN ratio. And the pre-treated algae alone, you can see that in case of the fresh algae with the cattle dung, biogas production was improved, but not as much as is in case of pre-treated one. And when I combine the pre-treatment with the cattle dung co-digestion, the methane production was significantly improved. So this was the journey which I started, and I work on improving the pre-treatment efficiency and improving the biogas production potential of the algal biomass. Yeah, so this is the highest methane yield, which was this, and which was around 62% higher than the control one. So this is, you can see that pre-treated with the cattle dung. Yeah, so this was about the challenges in the biogas production. The another challenge is, I think, do we have time to discuss? Anyone from organizing committee can comment?
Yes, sir. Once you finish your presentation, then we will discuss the topic.
Okay. Yeah. So the another challenge is that usually we grow algae in suspended culture, like photobioreactor or raised bed pond. The algae is grown in the media itself, and algae are having the size in micrometer. Okay, so what we can do, we cannot easily separate the algae from the liquid media. So I discuss about that we can grow the algae in the wastewater itself, but how we can separate the grown algae from the treated water? That was the another challenge in the process. So what I did, I selected that particular fungi which I use for the enzyme production. So same fungi, because the fungi grow in a filamentous like. Okay, so they grow in a mat type, and under the shaking they grow as a ball, large size balls which are easily settleable. Okay, so this property, I believe, I thought that it can be used for harvesting the algae, because I noticed that the fungi is having some charge, like positive charge on their surface, and the algae is having the negative charge. Okay, so I thought that due to the electrostatic interaction, there are possibility that these two can work together to get the biomass harvested. So I tested this process in the flask scale studies, and I found that within 6 hours, the fungal pellets which were grown earlier could harvest the algal biomass on their cell surface, and the liquid was clearly transparent, and all the biomass of the algae was settled. And that same process I tested at reactor scale. Again, within 6 hours of incubation, I found that we can easily harvest all the algae from the grown media, and we can easily decant the treated water. Okay, then because the algae was there and the fungi was there, this is the algae and fungi. So another idea came into my mind that this is the fungi which is producing the extracellular enzyme. So what I did, I stored the harvested biomass under control condition, dark condition, which are not supporting the growth of the algae, but the fungi could grow. And the microbes are very smart. They can sense that they have some substrate to eat. The fungi in the system could could sense that there is an algae which is having the carbon and can support their growth. So what they did, they started producing the enzyme, which you can see here from this graph. The enzyme started producing some extracellular enzyme. As a result of this, the biomass was pretreated. Within 2-3 days, the biomass was pretreated. Then I used this biomass in the biogas production assays, and I found that there is improvement in biogas production. You can see this is the A3, means the algal biomass alone harvested through some different method. The biogas production profile of that. And this is AF3, means algal fungal pellets which were stored for 3 days. So there was huge difference in the biogas production. So this was another the innovation which I discovered during the PhD. And this work has been recently patented, and the patent has been granted in 2019 itself, November. So I confirmed this process, and I found that with the help of fungi, we can treat the algal biomass and we can easily harvest the algal biomass. Okay, so that's why we can improve the biogas production from the algal biomass. So the overall, if we have wastewater, and if we know that how we can cultivate the fungi and the algae, we can club together them, and we can use some suitable co-substrate like cattle dung or the more organic solid waste from the municipals. So those can be taken up for the improved production of biogas utilizing the algal biomass. Yeah, again, based on the hypothesis that we can do the closed system development. So I actually did it. The hypothesis was validated. So what I did, I took the biomass, cultivated in bottles, I harvested it, and I used in the biogas production. Then liquid digested was recycled to the algae cultivation, and I found that biogas production was improved. Okay, based on the lab scale studies, I did some calculation for a plant which is having the biogas production of 1 meter cube per day, like a family type biogas plant. Okay, so for this we need around 3.15 kg of volatile solids on daily basis, for which we need algae cultivation facility of around 3.13 into 10⁻³ liter. Okay, and we can recycle the liquid digested. At the same time, we can have the rural sector wastewater or the sewage water, which should be added to the reactor, and additional water may be required if the wastewater is not sufficient. And with the help of this, we can treat the water, we can produce the biogas, and we can get the fertilizer. And the nutrient will be recycling again and again. And the CO2 from the atmosphere was being fixed during the algae cultivation. That was the added advantage. And that same model was then scaled up for a village having a population of 4,000 adult. And based on the calculation, I found that a village having 4,000 adult is having a potential to produce around more than 500 meter cube of methane per day, if we have adequate facility for algae cultivation. And the value were calculated, for example, based on the number of adult people, the rural sector water generation would be around 0.4 mld. So that water we are taking for algae cultivation. At the same time, we are recycling the liquid part from the anaerobic digestion to the cultivation facility, and we'll get the sufficient biomass to produce biogas. Okay, so this calculation showed that theoretically we can produce around more than 500 meter cube. If not 500 meter cube, practically if we can produce 200 meter cube methane per day, that will be sufficient to meet the energy requirement for that village. So later on, based on this some small funding from the Ministry of Rural Development, we started some project in one of the village of Haryana, Mubarakpur village of Haryana, but that was stopped in between because of the limited funding. So we could not explore the possibilities, but we have done something else, which I'll show you in the next slides. Okay, so you can see, this is the work which was carried out one of my PhD scholar who did his PhD jointly with IIT Delhi and with me. So the algal biomass, I hope you all know what is Hodgkiss Lake in Delhi. So Hodgkiss Lake, whenever you go, it will look in green, thick in green color. So this is because of huge growth of the algae, and this Hodgkiss Lake is receiving sewage treatment plant effluent, STP, which is rich in nutrients, because the conventional processes are not efficient to remove nutrients. For example, activated sludge process can remove the organic carbon, but cannot remove the most of the nutrient like nitrogen, phosphorus. So we noticed that there is a huge growth of the algae. So we collected that algal biomass. At the same time, we collected the algae, we cultivated the algae in the raceway pond in the lab itself, which we designed in our lab itself. And then both the biomass were harvested and taken with the cattle dung in a pilot scale plant. And the biogas production from this biogas plant, pilot scale plant, was monitored for more than 1 year. And we found that all the process parameters were stable within the range, and there was sufficient amount of biogas production from the this system. And initially we started with the cattle dung for a few days, then continuous mode. Initially it was for batch mode, then continuous mode for next 10 or 15 days. Then based on the mixed feed, having the algae and cattle dung, and the feed was the algae portion was step by step increased in the reactor. And finally it was tested for 1 year, and the biogas production was very stable from the system. This is the design of the reactor which was used, and you can see that the methane concentration, the biogas, which is very good. And initially there was some optimization period, then it was stabilized. You can see here. And obviously temperature plays a very crucial role, and particularly in Delhi and Roorkee region, the temperature fluctuation is very high with the season. So the temperature control should be there just to keep this graph in this range. But because of this low temperature after October or during the winter, the production of biogas was relatively low. And based on this, we are working on to link this type of technology with the biogas plant at family scale. And we are also looking for some industrial partner who those can test this particular integration at the dairy farm or wastewater treatment plants. Yeah, some of the work which I carried out during the PhD. So I have worked on the rural sector wastewater, sewage treatment water, milk processing water, livestock wastewater, slaughterhouse wastewater. So different wastewater were tested in the lab, and few of wastewater I work on, and few wastewater my colleague worked there. And those all try to grow the algae to produce the high biomass of algae. And this is the facility which were developed with the IIT Delhi. And some other attempts which I have made in the biogas production, like I have to improve the CY ratio. So this Algas is nothing but it's a term which was designed by my supervisor, Professor Anushi Malik, just to identify the gas produced from the algal biomass. So Algas is the biogas which is being produced from the algal biomass. So in similar way, like fungi can treat, I thought that that bacteria can also treat. So what I did, I collected the wastewater from the river, in the, some local river, and I grown that water, the algae under the, I incubated that water under the light conditions, and eventually after a few days, the algae started growing. And then I harvested this algal biomass, and I stored them in under natural conditions. Because of this, the pre-treatment occurred in the system itself. And the biogas production was improved, but not as much as in case of fungi, but there was significant improvement in the biogas production. Similarly, just to optimize the CYN ratio, I tried the algal food waste and cattle dung in a combination, and found that the biogas production can be improved. Okay, so these are few more studies which are related to my work in the algal biogas production. Yeah, so finally, I have ended with my technical discussion or the lecture. So I briefly introduce my team. Those are working in different aspects of the environment and bioprocess. Like Mohit is working on the nano particle mediated algal biofuel production. Mamta is working on the RO reject recycling for the algal based metabolite production. Ankita is working on the algae wastewater treatment for removal of pathogen from the wastewater. And Akansha and Pooja, those recently joined. So Akansha is working on the algae plus constructed wetland for wastewater treatment in rural sector. And we are looking to have some self-sustainable system in terms of if the algal produced during the treatment itself can be utilized for generation of energy to meet the requirement of energy in the process. And Pooja is working on the water body management, because in our department, there are two streams. One is the renewable energy, second one is the environment. And particularly we are focusing on the water bodies management. So we are having the MTech program on the environmental management of water, river and lake, which is a unique program which is being run in IIT Roorkee itself only. So this is in line with that. So she's working on the water bodies as well as bioremediation and greenhouse gas emission mitigation. Yeah, thank you very much. I believe there will be lots of questions from your side.
Okay, thank you, sir. Now I request topic is open for discussion, and I request participants to raise your questions. You can unmute yourself and ask question to sir directly, or you can put your question in the chat box. I will read on behalf of you. Sir, please, you can stop sharing your slides so that.
Yeah, please.
I request Dr. Bhavsar sir to ask his question.
Sir, I congratulate you for your innovative work. I also congratulate you for getting patent in this regard because this work is very new.
Yeah, thank you very much.
Which
This is very
algal
new.
species having? Yes, I have heard from you. This is very new, sir. Sir, which algal species having more potential?
So we tested different algae, including one procured one from the Chile, which was the Chlorella vulgaris, which was having very good potential in biogas production. But in my case, the native algal strain, which was isolated from the drain wastewater, and I found that this is not actually the algae. Initially, I was focusing it as algae, but that was the cyanobacteria, which is the blue-green algae. And molecularly, we identify it as Crococcus species. So Crococcus species was the one which was having the highest biogas production potential. And my entire work was based on that only. And third strain on which we have work is the Chlorella pionerosa. We obtain it from the NCL Pune. There is a culture collection from where we brought that culture. And the advantage of the Chlorella pionerosa is that it is having very high amount of carbohydrate content. So we are looking on if we can take the carbohydrate content for bioethanol production, and rest biomass we can produce, utilize for the biogas production.
Okay, thank you, sir. Now I request question from hall.
Thank you very much, sir. It's really wonderful hearing you.
So can you speak a little loudly because?
Yes, am I audible?
Yeah, please.
Thank you very much. It's really very interesting and new area to hear from you, and very challenging also. A lot of questions arise, but some of the questions you already shown there. But I'm interested in what are the possibilities of implementation in installation of algae biomass-based biogas power generation plant at village level. Is it possible, and what is the cost for that of that plant? And second thing is practically is it a user-friendly technology at village levels? That I want to know.
Yeah, thank you for your query. Yeah, obviously it is a user-friendly, but initially the people should be trained because it's a biological material, and some small disturbance can disturb the entire system. Okay, so in that case, yeah, it can be implemented at rural sector, and our aim when we work in the Mubarakpur village was just to ensure that it can be tested at village level. So obviously it can be implemented, but yeah, at least one skilled person is required who can maintain the problem and who can troubleshoot the issues. And the costing part, there are several views on the costing part. Particularly, we haven't done some costing estimation, but obviously when we are focusing on the wastewater treatment with the algal biomass production, the cost will be high. But initially, to build the raiseway pond, obviously there will be some expenditure. So we are not much aware of the calculation of the cost required for the initial setup, etc. But obviously there will be some cost.
Thank you very much.
Thank you, sir. Any other question?
Excuse me, sir. My question is, you explained here about algas. I would like to know how that algas is different from other gases like biogas, producer gas, in the form of thermal values and power generation values. How you will differentiate that gas?
So I think that you missed my explanation. So algas is nothing but it's a biogas, but just to differentiate it from the normal feedstock, this term was designed, but this is not very popular term, but I hope it will gain some popularity in near future. The algas is the biogas. Chemically it's methane and CO2 itself, but it is being produced from the algal biomass. So there is no different chemistry with the algas. Chemically it is same as biogas, mixture of CO2 and methane, but just to differentiate that this biogas is being produced from the algal biomass, we have given a new term like algas.
Sir, one more question. Actually, looking to the efficiency of the energy which is we get from this all these sources of biogas, whether it is from biomass, dung biomass, algal biomass, and the cost. What is the best and cheapest form of energy we will get?
So one thing is that biogas directly can be used for cooking. So this is the simplest way you can utilize the biogas. Otherwise, nowadays government is making efforts to make the like CNG we have for the transportation. Now there is a huge interest of from the government side to have the bio CNG or the compressed biogas. So chemically it is biogas, either you use any kind of substrate. Chemically it will be the methane only. So you are enriching the methane in the biogas up to 90 or 94%. So that can be used in the replacement or with the mixture with the CNG. And in Delhi region, I heard a news that there is a Inpras Gas Limited in the Delhi who is distributing the pipeline gas. So they were in plan to mix the compressed biogas with their CNG pipelines. So there are a few possibilities, and government is also supporting these kind of projects which are focusing on the compressed biogas, just to improve the use of bio CNG or CNG in the transport sector.
Thank you, sir. There is one question in the chat box, sir, from Mr. Prashant Raut, sir. He's asking, is any effect of heavy metal present in the wastewater on algal population?
Yeah, obviously, definitely there will be. I told you earlier that when we are cultivating the algae, the wastewater should be free from the toxic compound. Some strain are having the capabilities to bioremediate the heavy metals. So some strain can survive under the heavy metal load in the wastewater. But when you take the heavy metal containing algal biomass in the biogas production, obviously the biogas production will stop. Okay, there are only 1 or 2 papers those report that the heavy metal containing biomass can be used for biogas production. But up to my understanding, I do not believe if the heavy metal is there, obviously it will create some toxicity to the microorganisms which are present in the anaerobic digestion. So it's always good to remove the heavy metals prior you are using the wastewater for the algae cultivation.
Okay. Thank you, sir. Is there a question? Yes, sir.
There is one term you have shown that your student is working on bioremediation. So, what is the benefits? How we can deal the bioremediation in form of algae?
So bioremediation is a very broader term. Whenever we are using some biological material for treatment of wastewater, it is known as bioremediation. Okay, but the algae mediated treatment of wastewater is known as phycoremediation, which is a subclass of bioremediation. So when we are focusing on the any kind of living organism or the enzyme for the treatment, this is bioremediation. But particularly like for plant, we have the phytoremediation. Similarly for the algae, the term is phycoremediation.
Okay. Thank you, sir. There is another question in the chat box, sir. Which type of wastewater found good for getting energy?
Yeah, so we use the sewage wastewater, which was running from the hostels or the communities of the IIT itself. So those wastewater, because it was free from the contaminant like toxic contaminant, and we found that this is a very good substrate, the media for the algal growth. Secondly, I collected some water from the dairy farms, cattle dunks farm. So that water was very efficient, and the biogas production was up to 4 times higher in case of dairy liquid. Dairy farm wastewater. So obviously the cattle dung is available in the dairy farm, and if we can collect the wastewater generated from the dairy farm itself, so that will be the best way by which we can implement the algal biogas production at industrial scale.
Thank you, sir. Is there any question from the hall? Okay. Is there any question from the participants who are present at the online platform? You can raise your hand or you can put your question in the chat box. Okay. Thank you, sir. Now I request one of the participants from the hall to give feedback.
Yes, sir. Prajapati sir, on behalf of participants online and offline too, I am thank you, sir, for your very interesting work on challenges and opportunities of biogas production from algae biomass. And hopefully your work will become a breakthrough in the green energy production. And this, your lecture definitely opens new kind of window for us to work on and join hands with you for the contribution in the green energy production. Thank you very much once again.
Thank you very much.
Thank you, sir. Thank you very much for your nice and very interactive session. It was very informative presentation, sir. Your topic of presentation was very interesting, and I think you have enlightened our participants about how the herbal biomass can be used for the biogas production. So our participants are very happy, sir, meeting you and listening to you. So once again, thank you very much for joining us on this online platform, sir. Thank you.
Thank you very much. Still, I'll say that if there are some queries which I could not address here, and if some query will come in future in your mind, so everybody, all the participant or the organizing committee is free to drop me a mail. I will be happy to address that query. So thank you very much, the organizing team, for giving this opportunity to deliver a talk on this interesting topic.